The complement system acts in a concern with the antibodies, and it complements the action of the antibodies, and it consists of a group of a protein present in the plasma, and these proteins includes the complement components from the C1 to C9, mannose binding lectin, MBL, mannose binding lectin associated serine proteases, and the factor B, D, H, I, and others. Here the complement components are the inactive proenzymes, and these are converted into the active enzymes by a cascade of the reactions. And during the complement activation reaction, one complement protein acts on the next one, and so this is called the cascade. And the small peptide is split off from the second one, and the proenzyme is then converted into the active form of the enzyme. The smaller peptide splits off is denoted by the suffix A, and the larger fragment is denoted by the B. And the enzymatically active fragment is shown by the horizontal bar on it. Here we can see this the horizontal bar is above the A, means this is the active fragment. There are the three complement pathways and that we will read. Here the first is the classical complement cascade, another one is the lectin pathway, another is the alternate complement pathway. The classical complement cascade is activated by the antigen antibody binding, and the lectin and the alternate complement pathway don't require the antigen antibody interaction. The classical complement cascade, the classical complement cascade can be divided into the three phases: recognition phase, activation phase, and the membrane attack phase. In this recognition phase, a recognition unit participate in this phase. The recognition unit is made up of the C1, and it is C1q, C1r, and C1s. These are the subunit of the C1. In the activated phase, an activation unit participate in this phase. The other proteins are in this are the C1, C2, C3, and the C4. After that, the membrane attack phase is formed. In this, the membrane attack complex is formed, and the proteins that are included in this are made up of C5, C6, C7, C8, and the C9. In this, the first recognition phase, the binding of IgM or IgG to the bacterial antigen initiates the recognition phase. And one IgM or at least two IgG molecules are required for the initiation. And after the binding of the IgM or IgG to the antigen, it attracts the C1. Here we can see after the recognition phase, it is attracting the C1, and C1 having the C1q, R, and S. C1 is the complex made up of C1q, C1r, and C1s. C1q consists of the six identical subunits with a globular head and the long tail, and the tail combines to bind the two molecules each of the C1r and the C1s. This is the structure of the C1, and this C1q, C1r, S, and the C1r can be seen. C1 binds to the constant region of an antibody, and this binding causes a conformational change in the C1r. As a result, C1r cleaves and activates C1s, and this C1s is now been having the bar on its S, means it has been activated. Now the activation phase. In this activation phase, this C1 C4 is converted into the C4A and the C4B. And this C4B is the active form, and this converts the C2 protein into the C2B and the C2A. In this, the C2B is the active form, and this C2B is then combined with the C, C4B, and this is called the C3 complex. And this C3 is then C3 is protein acted by this and produce the C3A and the C3B. In this C3B active form acts on the C2 and split it into the C2B and the C2B activity form, and C4B and the C2B splits the C3 into the C3A and the C3B, and B form is the activated one. After that, the C3B splits the C5 into C5A and the C5B, and this C5B fragment and along with the C6, C7, C8 combine to form the membrane attack unit. The unit inserted in the plasma membrane of the target cells and attracts the C9, and the separate molecule of C9 then get inserted in the cell membrane of the target cell, and they are polymerized to form a transmembrane annular pores, and these pores leads to the lysis of the targeted cells. Here this can be seen the C9 protein poly making this membrane attack complex and making the pore on this cell membrane and rupturing it and making the holes in this pathogen and killing it. Another system is the lectin pathway. Lectin pathway, in this the complement pathway can also be activated by the mannose binding lectin. This is MBL. MBL is a protein synthesized by the liver and secreted in the circulation. MBL binds to mannose and some other carbohydrates of microbes. And binding of MBL to the microbial carbohydrates activates the MBL associated serine proteases, which is MASP. MASP activates the complement C2 and C4. The rest of the pathway is identical with that of the classical complement pathway. MASP activates the C2 and the C4. Here the mannose on the microbial surface can be seen on the pinkish color, and this MBL bind to the mannose on the surface of the microbes. After binding this, the MBL associated serine protease is activated, and this MBL associated serine proteases that convert the C2 into the two fragments C2B and the C2A and the C2-4B and C2-4A, and the C2B and the C4B are the activated form. MBL associated serine proteases here doing the process of the splitting out the C2 and the C4. Then this activated C2 and the C4 splits and activate the C3. Then they are doing? They are activating the C3, and the C3B is generated out. Another system here is the alternate complement pathway. In this pathway, the alternate complement pathway does not involve antigen-antibody interaction, and it does not require C1, C2 or C4, and it requires the rest of the component and the factors such as factor B and the factor D. According to the current view, C3 is slowly hydrolyzed when it contacts water, and this slow and the spontaneous hydrolysis occur continuously. If a foreign cell is not present, the active C3b bind to some um, self cell, and C3 is inactivated by the factor H and I present on the self cells. If a foreign cell is present, C3 bind to the foreign cells and C3 remain active because the foreign cells lack the factor H and the I. The protein, that is the factor B, bind to the C3b activated form to form a complex, and the factor D splits the factor B into capital B small b and the capital B small b mm, and b dash. Here the C3 complex is formed in the C3b b B dash split the C5 into C5A and the C5B dash. Here how this taking place is shown with the diagram with the C3. The C3 is is uh, C3 is converting into the C3B and the C3B capital form and the small form by splitting using the enzyme C3B B dash. After formation of the C5b, the pathway is similar to the classical complement cascade. Um, After the formation of the C5b, the pathway is similar to the classic complement cascade. The membrane attack complex is formed. The target cell is then lysed out. Here the C3, when after hydrolysis, it forms the C3A and C3B. And the C3B in this, the factor B acts and it forms the C3BB complex. And then after the factor D is act on it, it's formed the complex C3BBB complex. And this um, convert the C5 into C5A and C5B, and this C5B makes the membrane attack complex, and this membrane attack complex leads to the lysis of the targeted